Tonight's lecture is over Chapter 29, Development and Inheritance, and this chapter begins on page 1160 in your textbook. Both the genetic material inherited from parents, heredity, and normal development in the uterus environment play important roles in determining the homeostasis of a developing embryo and fetus and the subsequent birth of a healthy child. The embryonic period extends from fertilization through the 8th week. The first week of development is characterized by several significant events, including fertilization, cleavage of the zygote, blastocyst formation, and implantation. During fertilization, the genetic material from a haploid sperm and a haploid secondary oocyte merges into a single diploid nucleus. Of the 200 million sperm introduced into the vagina, fewer than 2 million reach the cervix of the uterus, and only about 200 reach the secondary oocyte. Fertilization normally occurs in the uterine tube within 12 to 24 hours after ovulation. Sperm can remain viable for about 48 hours after deposition in the vagina, although the secondary oocyte is viable for only about 24 hours after ovulation. Thus, pregnancy is most likely to occur if intercourse takes place during a 3-day window from 2 days before ovulation to 1 day after ovulation. Sperm swim from the vagina into the cervical canal with the whip-like movements of their tails. Their tails are termed flagella. The passage of sperm through the rest of the uterus and then into the uterine tube results mainly from contractions of the walls of these organs. The prostaglandins in semen are believed to stimulate the uterine motility at the time of intercourse and to aid in the movement of sperm through the uterus and into the uterine tube. Sperm that reach the vicinity of the oocyte within minutes after ejaculation are not capable of fertilizing it until about 7 hours later. Moving on to the next slide. This is figure 29.1 on page 1162, and this shows the selected structures and events in fertilization. For fertilization to occur, a sperm must first penetrate 2 layers of the corona radiata, the granulosa cells that surround the secondary oocyte, and the zona pellucida. The zona pellucida is the clear glycoprotein layer between the corona radiata and the oocyte's plasma membrane. Moving on to the next slide, this is the same figure on the same page, and we see a scanning electron micrograph of a sperm coming into contact with the secondary oocyte. The acrosome is a helmet-like structure that covers the head of the sperm, and it contains several enzymes. Acrosomal enzymes and strong tail movements help the sperm penetrate the cells of the corona radiata and come in contact with the zona pellucida. One of the glycoproteins in the zona pellucida, called ZP3, acts as a sperm receptor. Its binding to specific membrane proteins in the sperm head triggers the acrosomal reaction, and that releases the contents of the acrosome. The acrosomal enzymes digest a path through the zona pellucida, and the lashing sperm tail pushes the sperm onward. Although many sperm bind to the ZP3 molecules and undergo acrosomal reactions, only the first sperm to penetrate the entire zona pellucida and reach the oocyte's plasma membrane fuses with the oocyte. Moving on to the next slide. Once a sperm enters a secondary oocyte, the oocyte must complete meiosis II, and then it divides into a larger ovum, or a mature egg, and the smaller second polar body then fragments and disintegrates. The nucleus in the head of the sperm develops into the male pronucleus, and the nucleus in the fertilized ovum develops into the female pronucleus. After the male and female pronuclei form, they fuse, producing a single diploid nucleus, and this is a process known as syngamy. Thus, the fusion of the haploid pronuclei restores the diploid number of 46 chromosomes, and the fertilized ovum is now called a zygote. And I just want to mention this here because I frequently get questions on this, but dizygotic twins, or fraternal twins, are produced from the independent release of two secondary oocytes and the subsequent fertilization of each by different sperm. On the other hand, monozygotic twins develop from a single fertilized ovum, and they contain exactly the same genetic material and are always the same sex. So these are what we call identical twins. Monozygotic twins arise from the separation of the developing cells into two embryos, which in 99% of the cases occurs before 8 days have passed. Separations that occur later than 8 days are likely to produce conjoined twins. This is a situation in which the twins are joined together and share some of the same body structures. Moving on to the next slide. This is figure 29.2 on page 1163. After fertilization, rapid mitotic cell divisions of the zygote called cleavage take place. The first division of the zygote begins about 24 hours after fertilization and is completed about 6 hours later. Each succeeding division takes slightly less time. Moving on to the next slide, this is the same page and the same figure. By the second day after fertilization, the second cleavage is completed and there are 4 cells. And on this next slide, we see that by the end of the third day, there are 16 cells. The progressively smaller cells produced by cleavage are called blastomeres. Successive cleavages produce a solid sphere of cells called a morula. The morula is surrounded by the zona pellucida and is about the same size as the original zygote. Moving on to the next slide. By the end of the 4th day, the number of cells in the morula increases as it continues to move through the uterine tube toward the uterine cavity. When the morula enters the uterine cavity on day 4 or 5, glycogen-rich secretions from the glands of the endometrium of the uterus passes into the uterine cavity and enters the morula through the zona pellucida. This fluid is called uterine milk, along with the nutrients stored in the cytoplasm of the blastomeres of the morula. This provides nourishment to the developing morula. At the 32-cell stage, the fluid enters the morula, collects between the blastomeres, and reorganizes around a large fluid-filled cavity called the blastocyst cavity. Once the blastocyst cavity is formed, the developing mass is called a blastocyst. Though now it has hundreds of cells, the blastocyst is still about the same size as the original zygote. And this cartoon is also part of figure 29.2, and this shows the external view of the blastocysts at day 5. And moving on to the next slide, during the formation of the blastocyst, 2 distinct cell populations arise: the embryoblast and the trophoblast. The embryoblast, or innermost cell mass, is located internally and eventually develops into the embryo. The trophoblast is the outer superficial layer of cells that forms the sphere-like wall of the blastocyst. It will ultimately develop into the outer chorionic sac that surrounds the fetus and the fetal portion of the placenta. The placenta is the site of exchange of nutrients and waste between the mother and the fetus. And this cartoon is on page 1164. This is figure 29.3. The blastocyst remains free within the uterine cavity for about 2 days before it attaches to the uterine wall. At this time, the endometrium is in its secretory phase. About 6 days after fertilization, the blastocyst loosely attaches to the endometrium in a process called implantation. And this slide shows that implantation usually occurs in either the posterior portion of the fundus of the uterus or the body of the uterus. And the inner cell mass, as you can tell from this cartoon, orients towards the endometrium. And this cartoon takes us to page 1165, figure 29.4. Following implantation, the functional layer of the endometrium is known as the decidua. The decidua separates from the endometrium after the fetus is delivered, much as it does in normal menstruation. Different regions of the decidua are named based on their positions relative to the site of the implanted blastocyst. For instance, the basal decidua is the portion of the endometrium beneath the implanting embryo, and the capsular decidua is the portion of the endometrium that will cover the embryo after it implants. And the parietal decidua is the remaining modified endometrium that lines the non-involved areas and the rest of the uterus. And this brings us to page 1166, figure 29.5. And this cartoon summarizes the events associated with the first week of development. So at step 1, fertilization occurs in the uterine tube 12 to 24 hours after ovulation. At step 2, the first cleavage is completed about 30 hours after fertilization. At step 3, 3 to 4 days after fertilization, the morula is formed. At step 4, 4.5 to 5 days after fertilization, the blastocyst is formed. And then at step 5, implantation occurs about 6 days after fertilization. Moving on to the second week of development. About 8 days after fertilization, the trophoblast develops in 2 layers in the region of contact between the blastocyst and the endometrium. Like those of the trophoblast, cells of the embryoblast also differentiate into 2 layers around 8 days after fertilization, and this forms a hypoblast and an epiblast. Cells of the hypoblast and epiblast together form a flat disc referred to as the bilaminar embryonic disc. Soon after that, a small cavity appears within the epiblast and eventually enlarges to form the amniotic cavity. And this cartoon is on page 1167. This is figure 29.6 in your book. And this shows the principal events of the second week of development. Moving on to the next slide, as the amniotic cavity enlarges, a single layer of squamous cells forms a dome-like roof above the epiblast cells called the amnion. Thus, the amnion forms the roof of the amniotic cavity, and the epiblast forms the floor. Initially, the amnion overlies the bilaminar embryonic disc. However, as the embryonic disc increases in size and begins to fold, the amnion eventually surrounds the entire embryo. This creates the amniotic cavity that becomes filled with the amniotic fluid. The migrating columnar cells become squamous and then form a thin membrane referred to as the extraembryonic endoblast. This can also be referred to as the exocelamic membrane. Together with the hypoblast, the exocelamic membrane forms the wall of the umbilical vesicle, or yolk sac. On the 9th day after fertilization, the blastocyst becomes completely embedded in the endometrium. As the syncytiotropoblast expands, small spaces called trophoblastic lacunae develop within it. By the 12th day of development, the trophoblastic lacunae fuse to form larger, interconnecting spaces called vascular circles. And this cartoon is also part of figure 29.6, but this spans page 1167 and 1168. So as I've been saying, about 8 days after fertilization, the trophoblast develops into a syncytiotrophoblast and a cytotrophoblast, and the embryoblast develops into a hypoblast and an epiblast. Moving on to the next slide, about the 12th day after fertilization, the extraembryonic mesoderm develops. The mesoblast cells are derived from the umbilical vesicle and form a connective tissue layer called the mesenchyme around the amnion and umbilical vesicle. Soon, a number of large cavities develop in the extraembryonic mesoblast, which then fuse to form a single, larger cavity called the extraembryonic coelom. Moving on to the next slide, the extraembryonic mesoblast, together with the two layers of the trophoblast, forms the chorion. The chorion surrounds the embryo and later the fetus. Eventually, it becomes the principal embryonic part of the placenta, the structure for the exchange of materials between the mother and fetus. The chorion also protects the embryo and fetus from the immune responses of the mother in two ways. One, it secretes proteins that block the antibody production by the mother, and secondly, it promotes the production of T lymphocytes that suppress the normal immune response in the uterus. Finally, the chorion produces human chorionic gonadotrophin, an important hormone of pregnancy. Moving on to the next slide, the third embryonic week begins a 6-week period of very rapid development and differentiation. During the third week, the 3 primary germ layers are established and lay the groundwork for organ development in weeks 4 through 8. The first major event of the third week of development is gastrulation. This occurs about 15 days after fertilization. In this process, the bilaminar embryonic disc consisting of the epiblast and the hypoblast transforms into a trilaminar embryonic disc consisting of 3 layers, the ectoderm, the mesoderm, and the endoderm. These primary germ layers are the major embryonic tissues from which the various tissues and organs of the body will develop. Gastrulation involves the rearrangement and migration of cells from the epiblast. The first evidence of gastrulation is the formation of the primitive streak. This is a faint groove on the dorsal surface of the epiblast that elongates from the posterior to the anterior part of the embryo. And this cartoon appears on page 1169. This is figure 29.7, and this outlines the process of gastrulation. So remember, gastrulation involves the rearrangement and migration of cells from the epiblast. Moving on to the next slide. The primitive streak clearly establishes the head and tail ends of the embryo, as well as the right and left sides. At the head end of the primitive streak, a small group of epiblastic cells forms a rounded structure called the primitive node. Following the formation of the primitive streak, cells of the epiblast move inward below the primitive streak and detach from the epiblast. This is a process called invagination. Once the cells have invaginated, some of them displace the hypoblast, forming the endoderm. Other cells remain between the epiblast and the newly formed endoderm to form the mesoderm, and cells remaining in the epiblast then form the ectoderm. The ectoderm and endoderm are epithelia composed of tightly packed cells. The mesoderm is a loosely organized connective tissue. We sometimes call this the mesenchyme. And this brings us to table 29.1 on page 1170 of your book. This table summarizes the structures produced by the 3 primary germ layers. So please review this table on your own time. And this next slide is part of the same table on the same page. Please review this on your own time because I like to pull quiz and exam questions from tables. About 16 days after fertilization, mesodermal cells from the primitive node migrate toward the head end of the embryo and form a hollow tube of cells in the midline called the nodal chordal process. Then, by days 22 to 24, the nodal chordal process becomes a solid cylinder of cells called the notochord. This structure plays an extremely important role in induction. Induction is the process by which one tissue stimulates the development of an adjacent, unspecialized tissue into a specialized one. An inducing tissue usually produces a chemical substance that influences the responding tissue. The notochord induces certain mesodermal cells to develop into the vertebral bodies, and it also forms the nucleus pulposus of the intervertebral discs. And if we direct our attention to page 1170, figure 29.8, we can see the development of the nodal chordal process in this cartoon. The nodal chordal process develops from the primitive node and later becomes the notochord. Also, during the third week of development, two faint depressions appear on the dorsal surface of the embryo where the ectoderm and endoderm make contact but lack the mesoderm between them. The structure closest to the head is called the oral pharyngeal membrane. This structure breaks down during the fourth week to connect the mouth cavity to the pharynx and the remainder of the digestive canal. The structure closer to the tail end is called the cloacal membrane, and this degenerates in the seventh week to form the openings of the anus, urinary, and reproductive tracts. When the cloacal membrane appears, the wall of the umbilical vesicle forms a small vascularized outpouching called the allantois, and the allantois extends into the connecting stalk. And you can see that at figure 29.8b. In non-mammalian organisms that are enclosed in an amnion, the allantois is used for gas exchange and waste removal. Because of the role of the human placenta in these activities, the allantois is not a prominent structure in humans. Nevertheless, it does function in the early formation of blood and blood vessels and is associated with the development of the urinary bladder. And this cartoon is figure 29.9 on page 1172. This cartoon shows that the notochord also induces the development of the neural plate. And this next slide is part of the same cartoon on the same page, and this shows that the plate develops the neural fold as the lateral edges become more elevated. This slide, once again, is part of the same figure on the same page, and this shows the depressed mid-region of the fold becomes the neural groove. Again, this cartoon is part of the same figure, 29.9, and this shows as the neural folds approach each other and fuse, the neural tube is formed. Some of the ectodermal cells from the tube migrate to form several layers of cells called the neural crest. So these last four slides that I've shown you outline a process called neurulation, and this is the process by which the neural plate, the neural folds, and the neural tube form. Moving on to the next slide, at about 4 weeks after fertilization, the head end of the neural tube develops into 3 enlarged areas called primary brain vesicles. And the cartoon on this slide is back on page 539, figure 14.28. And this next slide takes us back to page 1172, figure 29.9, and we're viewing letter D on this figure. So by about the 17th day after fertilization, the mesoderm adjacent to the notochord and neural tube forms paired longitudinal columns of paraxial mesoderm. The mesoderm lateral to the paraxial mesoderm forms the cylindrical masses called the intermediate mesenchyme. The mesoderm lateral to the intermediate mesenchyme consists of a pair of flattened sheets called the lateral plate mesoderm. The paraxial mesoderm soon segments into a series of paired, cube-shaped structures called the somites. By the end of the 5th week, 42 to 44 pairs of somites are present. The number of somites that develop over a given period can be correlated to the approximate age of the embryo. Each somite differentiates into 2 distinct regions, a dermomitome and a sclerotome. The dermomitome further differentiates into a dermatome that will contribute to the formation of the subcutaneous tissue and dermis, and the myotome will give rise to all skeletal muscles of the trunk and limbs. The sclerotomes give rise to the vertebra and ribs. Moving on to the next slide, at the beginning of the 3rd week, angiogenesis, which is the formation of blood vessels, begins in the embryonic mesoblast in the umbilical vesicle, connecting stalk, and chorion. This early development is necessary because there is insufficient yolk in the umbilical vesicle and ovum to provide adequate nutrition for the rapidly developing embryo. Angiogenesis is initiated when mesodermal cells can differentiate into hemangioblasts. These hemangioblasts then develop into angioblasts, which aggregate to form isolated masses of cells referred to as blood islands. Moving on to the next slide, the heart forms from splotchnopleuric mesenchyme in the head end of the embryo on days 18 and 19. This region of mesodermal cells is called the cardiogenic mesenchyme. In response to induction signals from the underlying endoderm, the mesodermal cells form a pair of heart primordial tubes. The tubes then fuse to form a single heart tube. By the end of the 3rd week, the heart tube bends on itself, becoming S-shaped, and begins to beat. It then joins blood vessels in other parts of the embryo, connecting stalk, chorion, and umbilical vesicle to form the primitive cardiovascular system. And for this cartoon, we have to go back to page 759. This is figure 20.19, and this shows the development of the heart. The arrows within the structures indicate the direction of blood flow. Moving on to the next slide, this cartoon is figure 29.10 on page 1173. So as embryonic tissue invades the uterine wall, maternal uterine vessels are eroded and maternal blood fills spaces called trophoblastic lacunae. This happens within the invading tissue. By the end of the 2nd week of development, chorionic villi begin to develop. These finger-like projections consist of chorion that projects into the endometrial wall of the uterus. This slide is also the same figure and same page. By the end of the 3rd week, blood capillaries develop in the chorionic villi. This cartoon is also the same figure and same page. Blood vessels in the chorionic villi connect to the embryonic heart by way of the umbilical arteries and the umbilical vein through the connecting stalk, which will then eventually become the umbilical cord. The embryonic blood capillaries within the chorionic villi project into the trophoblastic lacunae, which unite to form the intervillous spaces. The intervillous spaces bathe the chorionic villi with maternal blood. As a result, maternal blood bathes the chorion-covered fetal blood vessels. Note, however, that maternal and embryonic blood vessels do not join, and the blood they carry does not normally mix. Instead, oxygen and nutrients in the blood of the mother's intervillous spaces diffuse across the cell membranes into the capillaries of the villi. Also, it should be noted here that waste products such as carbon dioxide diffuse in the opposite direction. Moving on to the next slide, placentation is the process of forming the placenta. As I've said before, the placenta is the site of the exchange of nutrients and waste between the mother and embryo or fetus. The placenta also produces hormones needed to sustain the pregnancy. The placenta is unique because it develops from 2 separate individuals, the mother and the embryo. And that brings us to figure 29.11 on page 1174. By the beginning of the 12th week, the placenta has two distinct parts. The fetal portion, formed by the chorionic villi of the chorion, and the maternal portion, formed by the basal layer of the endometrium of the uterus. When fully developed, the placenta is shaped like a pancake. Functionally, the placenta allows oxygen and nutrients to diffuse from maternal blood into fetal blood, while carbon dioxide and waste diffuse from fetal blood into the maternal blood. And this next slide is also part of figure 29.11. This shows us the fetal surface of the placenta. And this slide shows us figure 29.12 on page 1176. The 4th through 8th weeks of development are significant in embryonic development because all major organs appear during this time. The term organogenesis refers to the formation of body organs and systems. By the end of the 8th week, all of the major body systems have begun to develop, although their functions, for the most part, are minimal. Organogenesis requires the presence of blood vessels to supply the developing organs with oxygen and other nutrients. However, recent studies suggest that blood vessels play a significant role in organogenesis even before blood begins to flow within them. The endothelial cells of blood vessels apparently provide some type of developmental signal, either a secreted substance or a direct cell-to-cell interaction that is necessary for organogenesis. During the 4th week after fertilization, the embryo undergoes a very dramatic change in size and shape. It nearly triples in size. It essentially is converted from a flat, two-dimensional trilaminar embryonic disc to a three-dimensional cylinder. This is a process called embryonic folding. The embryonic cylinder consists of endoderm in the center, or the gut, the ectoderm on the outside, the epidermis, and the mesoderm in between. The main force responsible for embryonic folding is the different rates of growth of the various parts of the embryo, especially the rapid longitudinal growth of the nervous system. Folding in the median plane produces a head fold and a tailed fold. Folding on the horizontal plane results in two lateral folds. Overall, due to the foldings, the embryo curves into a C shape. The head fold brings the developing heart and mouth into their eventual adult positions. The tail fold brings the developing anus into its eventual adult position. And this slide is part of the same figure, same page. The lateral folds form as the lateral margins of the trilaminar embryonic disc bend ventrally. As they move toward the midline, the lateral folds incorporate the dorsal part of the umbilical vesicle into the embryo as the primitive gut. This is the forerunner of the digestive canal. The primitive gut differentiates into the anterior foregut, an intermediate midgut, and a posterior hindgut. Again, this slide is part of the same figure and the same page. In a developing embryo, the last part of the hindgut expands into a cavity called the cloaca. On the outside of the embryo is a small cavity in the tail region called the proctodeum. And this slide is the same figure, same page. Separating the cloaca from the proctodeum is the cloacal membrane. During embryonic development, the cloaca divides into a ventral urogenital sinus and a dorsal anorectal canal. As a result of tail folding, the cloacal membrane moves downward and the urogenital sinus, anorectal canal, and proctodeum move closer to their final positions. When the cloacal membrane ruptures during the 7th week of development, the urogenital and anal openings are created. And this slide begins figure 29.13 on page 1177. So in addition to embryonic folding, development of the somites, and the development of the neural tube, 4 pairs of pharyngeal arches begin to develop on each side of the future head and neck regions. And this happens during the 4th week. These four paired structures begin their development on the 22nd day after fertilization and form swellings on the surface of the embryo. Each pharyngeal arch consists of an outer covering of ectoderm and an inner covering of endoderm, with mesoderm in between. Within the pharyngeal arch, there is an artery, a cranial nerve, cartilaginous skeletal rods that support the arch, and skeletal muscle tissue that attaches and moves the cartilage rods. On the ectodermal surface of the pharyngeal region, the pharyngeal arch is separated by a groove called the pharyngeal groove. The pharyngeal grooves meet corresponding balloon-like outgrowths of the endodermal pharyngeal lining called the pharyngeal pouches. Where the pharyngeal groove and pouch meet to separate the arches, the outer ectoderm of the cleft contacts the inner endoderm of the pouch, and there is no mesoderm in between them. And this cartoon is also part of figure 29.13 on page 1177. And this shows the pharyngeal pouches that meet the pharyngeal clefts. And remember, at this point, there is no mesoderm. Moving on to the next slide. By the middle of the 4th week, the upper limbs begin their development as outgrowths of the mesoderm covered by ectoderm. This is called the upper limb buds, and by the end of the 4th week, the lower limb buds develop. The heart also forms a distinct projection on the ventral surface of the embryo called the heart prominence. Also, at the end of the 4th week, the embryo has a distinctive tail. This is figure 29.14, and you can see this on page 1178 of your textbook. During the 5th week of development, there is a very rapid development of the brain, so the growth of the head is considerable. By the end of the 6th week, the head grows even larger relative to the trunk, and the limbs show substantial development. In addition, the neck and trunk begin to straighten, and the heart is now 4-chambered. And this slide is the same figure on page 1178. By the 7th week, the various regions of the limbs become distinct, and the beginnings of the digits begin to appear. And this slide too is the same figure, 29.14. So at the start of the 8th week, the digits of the hands are short and webbed, the tail is shorter but still visible, the eyes are open, and the auricles of the ear are visible. By the end of the 8th week, all regions of limbs are apparent. The digits are distinct and no longer webbed due to the removal of cells via apoptosis. Also, the eyelids come together and may fuse. The tail disappears, and the external genitals begin to differentiate. So this slide is also figure 29.14. During fetal development, which starts in the 9th week until birth, tissues and organs that developed during the embryonic period grow and differentiate. Very few new structures appear during the fetal period, but the rate of body growth is remarkable, especially during the second half of intrauterine life. And this slide is table 29.2 on page 1179, and this gives you a summary of the changes during embryonic development. Please go through this table on your own time. And this slide is part of the same table. And this slide is also part of the same table, but now it's summarizing the changes during fetal development. Remember, fetal development begins at 9 to 12 weeks. And this next slide is part of the same table, same page. Please make sure you review this. This slide, part of the same table. And this slide is number 4 of 5, of course, part of the same table and the same page. And here is slide 5 of 5, and this part of the table actually appears on page 1180. So moving on to teratogens on page 1180, exposure of a developing embryo or fetus to certain environmental factors can damage the developing organism or even cause death. A teratogen is any agent or influence that causes developmental defects in the embryo. Because the placenta is not an absolute barrier between the maternal and fetal circulations, any drug or chemical that is dangerous to an infant should be considered potentially dangerous to the fetus when given to the mother. Alcohol is by far the number one fetal teratogen. Intrauterine exposure to even a small amount of alcohol may result in fetal alcohol syndrome. This is one of the most common causes of mental retardation and the most common preventable cause of birth defects in the United States. Other teratogens include viruses, industrial chemicals, some hormones, antibiotics, and cocaine, just to name a few. Moving on to the next slide. Cigarette smoking during pregnancy has also been implicated as a cause of low infant birth weight, cardiac abnormalities, encephaly, and a higher infant and fetal mortality rate. Also, ionizing radiation in many forms is also teratogenic. For instance, the exposure of the mother to X-rays or radioactive isotopes during pregnancy may cause microcephaly, mental retardation, and skeletal deformities. And this slide talks about prenatal diagnostic tests. During pregnancy, several medical tests are used to detect fetal abnormalities, genetic disorders, and well-being. If there is a question about the normal progress of a pregnancy, fetal ultrasonography may be performed. By far the most common use of diagnostic ultrasound is to determine a more accurate fetal age when the date of conception is unclear. It is also used to confirm pregnancy, evaluate fetal viability and growth, determine fetal position, identify multiple pregnancies, identify fetal maternal abnormalities, and serve as an adjunct to special procedures such as amniocentesis. Moving on to the next slide, amniocentesis involves withdrawing of some of the amniotic fluid that bathes the developing fetus and analyzing the fetal cells and dissolved substances. It is used to test for the presence of certain genetic disorders such as Down syndrome, hemophilia, Tay-Sachs, sickle cell, and certain muscular dystrophies. The test is usually done at 14 to 18 weeks of gestation. And regarding the execution of this test, there is a needle that is used to collect the amniotic fluid, and this needle is guided by ultrasound to avoid damage to the fetus or umbilical cord. And this cartoon appears on page 1182 of your book. This is figure 29.15. And this shows a cartoon of the amniocentesis test. And in the next slide, we talk about chorionic villi sampling. And this is where a catheter is guided through the vagina and cervix of the uterus and then advanced to the chorionic villi under ultrasound guidance. The goal of this test is to identify the same genetic defects as seen with amniocentesis. And this procedure may also be done through the abdominal wall as with amniocentesis. And this cartoon is letter B of figure 29.15. Where it's showing an example of how the chorionic villi sampling may occur. Moving on to the next slide. Currently, chorionic villi testing and amniocentesis are the only useful ways to obtain fetal tissue for prenatal testing of gene defects. While these invasive procedures pose relatively little risk when performed by experts, much work has been done to develop non-invasive prenatal tests which do not require the penetration of any embryonic structure. The goal is to develop accurate, safe, more efficient, and less expensive tests. The first such test developed was the maternal alpha-fetoprotein test, or the AFP test. In this test, the mother's blood is analyzed for the presence of AFP. AFP is a protein synthesized in the fetus that passes into the maternal circulation. The highest levels of AFP occur during weeks 12 through 15 of the pregnancy. A high level of AFP after week 16 usually indicates that the fetus has a neural tube defect. Moving on to maternal changes during pregnancy. This is on page 1183. During the first 3 to 4 months of pregnancy, the corpus luteum in the ovary continues to secrete progesterone and estrogens, which maintain the lining of the uterus during pregnancy and prepare the mammary glands to secrete milk. From the 3rd month through the remainder of the pregnancy, the placenta itself provides the high levels of progesterone and estrogens that are required. As noted previously, the chorion of the placenta secretes human chorionic gonadotropin into the blood. This is figure 29.16 on page 1183, and this shows the hormones during pregnancy. So the corpus luteum produces progesterone and estrogens during the first 3 to 4 months of pregnancy, after which time the placenta assumes its function. Relaxin, a hormone produced first by the corpus luteum of the ovary and later by the placenta, increases the flexibility of the pubic symphysis and ligaments of the sacroiliac and sacrococcygeal joints and helps dilate the uterine cervix during labor. Both of these actions ease the delivery of the baby. A third hormone produced by the chorion of the placenta is the human chorionic somatomammotrophin, or HCS. The rate of HCS secretion increases the proportion to the placental mass, reaching the maximum levels after 32 weeks and remaining relatively consistent after that. It is thought to help prepare the mammary glands for lactation, enhance maternal growth by increasing protein synthesis, and regulate certain aspects of metabolism in both the mother and the fetus. For example, HCS decreases the use of glucose by the mother and promotes the release of fatty acids from her upper adipose tissue, making more glucose available to the fetus. And the hormone most recently found to be produced by the placenta is corticotropin-releasing hormone. In non-pregnant people, this is secreted only by neurosecretory cells in the hypothalamus. CRH is now thought to be part of the clock that establishes the timing of birth. Secretion of CRH by the placenta begins at about 12 weeks and increases enormously towards the end of pregnancy. Women who have higher levels of CRH earlier in pregnancy are more likely to deliver prematurely. Those who have low levels are more likely to deliver after the due date. CRH from the placenta has a second important effect, and that is that it increases the secretion of cortisol, which is needed for the maturation of the fetal lungs and the production of surfactant. A surfactant is a substance which tends to reduce the surface tension of a liquid in which it is dissolved. So moving on to the next slide, this is also part of figure 29.16, but it's continued on page 1184. And this just shows the blood levels of hormones during pregnancy. So HCG levels peak at about the 9th week of pregnancy. And the chorion secretes estrogens after the first 3 or 4 weeks of pregnancy and progesterone by the 6th week of pregnancy. Moving on to the next slide, near the end of the 3rd month of pregnancy, the uterus occupies most of the pelvic cavity. As the fetus continues to grow, the uterus extends higher and higher into the abdominal cavity. Toward the end of a full-term pregnancy, the uterus fills nearly the entire abdominal cavity, reaching above the costal margin nearly to the xiphoid process of the sternum. It pushes the maternal intestines, liver, and stomach superiorly. It elevates the diaphragm, widens the thoracic cavity, and puts pressure on the stomach that may force the stomach contents superiorly into the esophagus, resulting in heartburn. In the pelvic cavity, compression of the ureters and urinary bladder also can occur. And this cartoon appears on page 1185. This is figure 29.17. This shows the normal fetal location and position at the end of a full-term pregnancy. The gestation period is the time interval from fertilization till birth, and in humans this takes about 38 weeks. Exercise in pregnancy begins on page 1186. Only a few changes in early pregnancy affect the ability to exercise. A pregnant woman may tire more easily than usual, or morning sickness may interfere with the regular exercise routine. As the pregnancy progresses, weight is gained, posture changes, and more energy is needed to perform activities and certain maneuvers, such as sudden stopping, changes in direction, and rapid movements. These types of maneuvers become more difficult to execute. In addition, certain joints, especially the pubic symphysis, may become less stable in response to an increased level of the hormone relaxin. As compensation, many mothers-to-be walk with widely spread legs in a shuffling motion. And moving on to labor. Labor is the process by which the fetus is expelled from the uterus through the vagina, also referred to as giving birth. The onset of labor is determined by complex interactions of several placental and fetal hormones. Because progesterone inhibits uterine contractions, labor cannot take place until the effects of progesterone are diminished. Toward the end of gestation, the levels of estrogens in the mother's blood rise sharply. This produces changes that overcome the inhibiting effects of progesterone. Also, control of contractions during labor occurs via a positive feedback cycle. Moving on to the next slide, uterine contractions occur in waves that start at the top of the uterus and move downward, eventually expelling the fetus. True labor begins when the uterine contractions occur at regular intervals, and this usually produces pain. As the interval between contractions shortens, the contractions intensifies. Another symptom of true labor in some women is the localization of pain in the back that is intensified by walking. The most reliable indicator of true labor is a dilation of the cervix and the quote-unquote show. This is a discharge of blood containing mucus into the cervical canal. In false labor, pain is felt in the abdomen at regular intervals but does not intensify, and walking does not alter it significantly. There is no quote-unquote show and no cervical dilation. So true labor can be divided into 3 stages. The first stage is dilation. The time from the onset of labor to the complete dilation of the cervix is the stage of dilation. This stage, which typically lasts 6 to 12 hours, features regular contractions of the uterus, usually a rupturing of the amniotic sac and complete dilation of the cervix. If the amniotic sac does not rupture spontaneously, it is ruptured intentionally. The second stage is a stage of expulsion. The time can take 10 minutes to several hours from complete cervical dilation to the delivery of the baby. And finally, the placental stage. In the placental stage, the time usually takes about 3 to 5 minutes to an hour after delivery until the placenta, or quote-unquote afterbirth, is expelled by powerful uterine contractions. These contractions also constrict blood vessels that were torn during delivery, reducing the likelihood of hemorrhage. And this cartoon is on page 1187. This is figure 29.18. And this just goes through the true stages of labor, which I outlined on the previous slide. Moving on to the next slide. Following the delivery of the baby and placenta is a 6-week period during which the maternal reproductive organs and physiology return to the pre-pregnancy state. This period is called the puerperium. Through a process of tissue catabolism, the uterus undergoes a remarkable reduction in size called involution. For 2 to 4 weeks after delivery, women have a discharge called lochia, which consists initially of blood and later of serous fluid derived from the former site of the placenta. Moving on to the adjustments of the infant after birth. During pregnancy, The embryo and later fetus is totally dependent upon the mother for its existence. The mother supplies the fetus with oxygen and nutrients, eliminates its carbon dioxide and other wastes, protects it against shock and temperature changes, and provides antibodies that confer protection against certain harmful microbes. At birth, a physiologically mature baby becomes much more self-supporting, and the newborn's body systems must make various adjustments. The reason that the fetus depends entirely on the mother for obtaining oxygen and eliminating carbon dioxide is that the fetal lungs are either collapsed or partially filled with amniotic fluid. The production of surfactant begins by the end of the 6th month of development. Because the respiratory system is fairly well developed at least 2 months before birth, premature babies delivered at 7 months are able to breathe and cry. After delivery, the baby's supply of oxygen from the mother ceases and any amniotic fluid in the fetal lungs is absorbed. Because carbon dioxide is no longer being removed, it builds up in the blood. A rising carbon dioxide level stimulates the respiratory center in the medulla oblongata. This causes the respiratory muscles to contract and the baby to draw his or her first breath. The respiratory rate at birth is 45 breaths per minute, and this will drop to the normal 12 breaths per minute within 2 weeks. Moving on to the next slide. After the baby's first inspiration, the cardiovascular system must make several adjustments. One is the closure of the foramen ovale between the atria of the fetal heart, which occurs shortly after birth. This diverts deoxygenated blood to the lungs for the first time. The foramen ovale is closed by 2 flaps of the septal heart tissue that fold together and permanently fuse. The remnant of the foramen ovale is the fossa ovalis. Once the lungs begin to function, the ductus arteriosus shuts off due to the contractions of smooth muscles in its wall, and it becomes the ligamentum arteriosum. After the umbilical cord is tied off and severed, the blood no longer flows through the umbilical arteries. They fill with connective tissue and become the medial umbilical ligaments. The umbilical vein then becomes the ligamentum teres of the liver. And this slide takes you back to figure 21.31 in chapter 21 on page 836. This shows the fetal circulation and the changes that occur at birth. The gold boxes between parts A and B describe the fate of certain fetal structures once postnatal circulation is established. Moving on to the physiology of lactation. Lactation is the production and ejection of milk from the mammary glands. A principal hormone in promoting milk production is called prolactin. This hormone is secreted from the anterior pituitary gland. Even though prolactin levels increase as the pregnancy progresses, no milk production occurs because progesterone inhibits the effects of prolactin. After delivery, the levels of estrogens and progesterone in the mother's blood is decreased, and the inhibition on prolactin is removed. The principal stimulus in maintaining prolactin secretion during lactation is the sucking action of the infant. Suckling initiates nerve impulses from stretch receptors in the nipples to the hypothalamus. The impulses decrease hypothalamic release of prolactin-inhibiting hormone and increase the release of prolactin-releasing hormone, so more prolactin is released by the anterior pituitary. Furthermore, oxytocin causes the release of milk into the mammary ducts via the milk ejection reflex. And this is page 1190, figure 29.19 in your book. And this shows that the milk ejection reflex is a positive feedback cycle. Oxytocin stimulates the contraction of myoepithelial cells in the breasts, which squeezes the glandular and duct cells, and this causes the milk ejection. Moving on to the next slide, there are many benefits associated with breastfeeding an infant. One is the chemical composition of the mother's milk is ideal for the baby's brain development, growth, and digestion. Also, several types of white blood cells are in the milk, and this is good for the baby's immunity. Furthermore, there are antibodies present in the milk. breastfeeding supports optimal infant growth, and breastfeeding leads to a reduction in several diseases. Moving on to inheritance on page 1191. Inheritance is the passage of hereditary traits from one generation to the next. It is the process by which you acquired your characteristics from your parents and may transmit some of your traits to your children. The branch of biology that deals with inheritance is called genetics. The area of healthcare that offers advice on genetic problems is called genetic counseling. You have already learned that the nuclei of all human cells except the gametes contain 23 pairs of chromosomes. One chromosome in each pair came from the mother and one came from the father. Each of these two homologs contains genes that control the same traits. If one chromosome of the pair contains a gene for body hair, for example, the homolog will contain a gene for body hair in the same position. Alternate forms of a gene that code for the same trait at the same location on homologous chromosomes are called alleles. One allele of the previously mentioned body hair gene might code for the coarse hair, and another might code for fine hair. A mutation is a permanent heritable change in an allele that produces a different variant of the same trait. The relationship of genes to heredity can be illustrated by examining the alleles involved in the disorder called phenylketonuria. People with PKU are unable to manufacture the enzyme phenylalanine hydroxylase. This enzyme is required to break down phenylalanine. Phenylalanine is an amino acid, and if you cannot break this amino acid down, it is toxic to the nervous system. And without treatment, this can cause severe intellectual disabilities. Moving on to the next slide. The genotype is the actual genetic makeup relating to a trait. An allele that dominates or masks the presence of another allele is called a dominant allele, and this will be represented by an uppercase letter. The allele whose presence is completely masked is the recessive allele, and this will be represented by a lowercase letter. And the phenotype is the physical expression of the genotype. So in other words, the phenotype is the allele or the trait that penetrates the observable trait. So this is figure 29.20 on page 1191. So this figure shows the possible combinations of gametes from two parents who each have one dominant P allele and one recessive P allele. And this type of diagram is called a Punnett square. In constructing a Punnett square, the possible pattern of alleles in sperm are written on the left side, and the possible maternal alleles in the ova are written on the top. The four spaces on the chart show how the alleles can combine in zygotes formed by the union of these sperm and ova to produce three different combinations of genes or genotypes. Those include capital P, capital P lowercase p, or lowercase p. So when there are two letters of the same case, we would term that homozygous. And when there are two letters of differing cases, in other words, capital P, lowercase p, we call that heterozygous. So when you have a capital P, your homozygous dominant, and lowercase p, your homozygous recessive. So what we can glean from this figure, from this Punnett square, is that this couple, when they produce offspring, the chances are that 25% of that offspring will have a homozygous dominant genotype. 50% of these offspring will be heterozygous for this genotype, and another 25% of the offspring that this couple could produce will have the homozygous recessive genotype. So any offspring that are homozygous dominant or heterozygous will not have PKU, but any offspring that are homozygous recessive will suffer from the disorder PKU. So this table 29.3 is on page 1192, And this just shows some of the selected heredity traits in humans, and it shows whether they're dominant or recessive. And this next slide is a continuation of the same table. Please review this table on your own time. So moving on to the next slide, the figure that I showed you at 29.20 was of simple dominant recessive inheritance. However, most patterns of inheritance do not conform to the simple dominant recessive inheritance that we described in figure 29.20. The phenotypic expression of a particular gene may be influenced not only by which alleles are present, but also by other genes and by the environment. Most inherited traits are influenced by more than one gene, and, to complicate matters, most genes can influence more than one trait. Variations on dominant recessive inheritance include incomplete dominance, multiple allele inheritance, and complex inheritance. In incomplete dominance, neither member of a pair of alleles is dominant over the other, and the heterozygote has a phenotype intermediate between the homozygous dominant and the homozygous recessive phenotypes. An example of incomplete dominance is sickle cell disease. So this next slide is figure 29.21 on page 1193, and this shows the inheritance of sickle cell disease. People with the homozygous dominant genotype, HbA form normal hemoglobin. Those with the homozygous recessive genotype, HbS, have sickle cell disease and severe anemia. Although they are usually healthy, those with the heterozygous genotype, HbS, have minor problems with anemia because half of their hemoglobin is normal and half is not. Heterozygotes are carriers, and they are said to have the sickle cell trait. Moving on to the next slide, although a single individual inherits only 2 alleles for each gene, some genes may have more than 2 alternate forms. This is the basis for multiple allele inheritance, or codominance. And this is figure 29.22 on page 1193, and this shows the 10 possible combinations of prenatal ABO blood types and the blood types their offspring could inherit. For each possible set of parents, the blue letters represent the blood types their offspring could inherit. Notice that both IA and IB are inherited as dominant alleles, and I is inherited as a recessive allele. Because an individual with type AB blood has characteristics of both type A and type B red blood cells expressed in the phenotype, the alleles IA and IB are said to be codominant. In other words, both genes are expressed equally in the heterozygote. Depending on the parental blood types, different offspring may have blood types different from each other. Moving on to complex and polygenic inheritance, most inherited traits are not controlled by one gene, but instead by the combined effects of 2 or more genes. This is a situation referred to as polygenic inheritance, or the combined effects of many genes and environmental factors. This is a situation referred to as complex inheritance. Examples of complex traits include skin color, hair color, eye color, height, metabolic rate, and body build. Skin color is a good example of a complex trait. It depends on environmental factors such as sun exposure and nutrition, as well as several genes. Suppose that skin color is controlled by 3 separate genes, A, B, and C. Then a person with the genotype of all dominant A, B, and C's is very dark-skinned, and an individual with the genotype of recessive A, B, and C's is very light-skinned, and a person with the genotype that's heterozygous for each one of those letters has an intermediate skin color. Parents having an intermediate skin color may have children with very light, very dark, or intermediate skin color. And when we move on to figure 29.23 on page 1194, we see that the P generation, or the parental generation, is the starting generation, and that the F1 generation is the first filial generation. This is produced by the P generation, and the F2 generation is the second filial generation, and this is produced by the F1 generation. Notice that in the P generation, parents are homozygous recessive and homozygous dominant, and in the F1 generation, the children are heterozygous for all the genes that make up this trait. And then when we get into the F2 generation, there's a range of possibilities that can occur with the heterozygous parents of the F1 generation. Moving on to the next slide, the 46 human chromosomes, or 23 pairs of chromosomes, are identified by their size, shape, and staining pattern. A karyotype is an entire set of chromosomes arranged in decreasing size order and according to the position of the centromere. Moving on to figure 29.24 on page 1194, we see a human karyotype showing autosomes and sex chromosomes. The white circles are the centromeres, and the sex chromosomes are in blue. Moving on to the next slide, the 23 pairs of human chromosomes include 22 pairs of autosomes and 1 pair of sex chromosomes. And the sex chromosomes are termed X and Y. Males are what we call hemizygous because they have an X and a Y chromosome. Females have 2 X chromosomes, and they can be either homozygous or heterozygous for their sex chromosomes. In the case of the female with 2 X chromosomes, one becomes automatically inactivated, and this is called X inactivation. And this brings us to figure 29.25 on page 1195, and this shows us how sex determination can be inherited. So moving on to the next slide, in addition to determining the sex of the offspring, the sex chromosomes are responsible for the transmission of several non-sexual traits. Many of the genes for these traits are present on X chromosomes but are absent from Y chromosomes. This feature produces a pattern of heredity called sex-linked inheritance that is different from the patterns already described. One example of sex-linked inheritance is red-green color blindness. And in figure 29.26 on page 1195, we see an example of the inheritance of red-green color blindness. The gene for red-green color blindness is a recessive one designated lowercase c. Normal color vision, designated capital case C, dominates. The capital C, lowercase c genes are located only on X chromosomes, so the ability to see colors depends entirely on the X chromosomes. Because males do not have a second X chromosome that could mask the trait, all males with an X lowercase c gene will be red-green colorblind. This disorder is rare in females because females who have 2 X lowercase c genes are red-green colorblind. This can only result from the mating of a colorblind male and a colorblind or carrier female. Moving on to the next slide, female infertility, or the inability to conceive, occurs in about 10% of all women of reproductive age in the United States. Female infertility may be caused by ovarian disease, obstruction of the uterine tubes, or conditions in which the uterus is not adequately prepared to receive a fertilized ovum. On the other hand, male infertility or sterility occurs in about 10% of males in the United States and is defined as an inability to fertilize a secondary oocyte. However, this does not imply erectile dysfunction. Male fertility requires the production of adequate quantities of viable, normal sperm by the testes, unobstructed transport of sperm through the ducts, and satisfactory deposition in the vagina. The seminiferous tubules and the testes are sensitive to many factors: X-rays, infections, toxins, malnutrition, and higher than normal scrotal temperatures. Those scenarios may cause degenerative changes and produce male sterility. An abnormality that is present at birth, and usually before, is called a congenital defect. Such defects occur during the formation of structures that develop during the period of organogenesis, the 4th through 8th weeks of development, when all the major organs appear. During organogenesis, stem cells are establishing the basic patterns of organ development, and it is during this time that developing structures are very susceptible to genetic and environmental influences. Down syndrome is a disorder characterized by 3 rather than 2 copies of chromosome 21. Sometimes this is called trisomy 21. Down syndrome is characterized by mental retardation, retarded physical development, distinctive facial features, kidney defects, suppressed immune system, and malformations of the heart, ears, hands, and feet. Sexual maturity is rarely attained, and life expectancy is shorter. And that concludes tonight's lecture.